Today we're going to step back a little bit, just briefly away from the technicalities of coding and try to think like scientists who have hypotheses and design experiments, and also to think as a data literate person, right? Scientists are not always data literate, but you are going to be both. So as you're designing this experiment, to have the concept of the statistics and the machine reading your data kind of in the back of your head. So we're going to design an experiment together. This is going to be super exciting. Let me pull up Word document. Okay, I'm going to share my screen. Okay. The effect of soil moisture. How about we call that water content? Water content on 3 species of plants in conjunction with a new agricultural treatment. All right. So let's design an experiment to measure this. So give me your thoughts. And let's say we're doing this in like a greenhouse. Okay. Greenhouse. What are my independent variables here?
Independent variables will be the 3 species of plants and then probably a control.
Okay, so
well, and amount of water and treatment. We're giving it a treatment too.
Okay, so the ag treatment and water.
Yes, I agree with that.
Anyone have thoughts? Your silence implies that you agree. Okay, great. Dependent variable, or voles.
You could measure the mass of the plants or space between each nodes.
Okay.
You can measure their oxygen intake for their energy use.
Or energy use or CO2 intake. Okay. So what's this going to look like in a data frame? The question I'm asking, here's the jargon version.
Well,
what are the levels? What are the levels of our independent variables? You got plants. So that'll be like plant A, plant B, plant C. Okay. What about the ag treatment?
Just present or absent, right?
Okay, so that's true, false.
Well, you'd probably want another ag treatment known to be effective to compare to, maybe. So maybe just a control too.
The control in this case would actually be no ag treatment. So to keep this a bit simpler, let's do whether it's applied or not. So, okay. So we'll stick with true, false, false being the control. And what about water?
We'd probably say liters or something.
All right. So liters of water added doesn't necessarily lead to soil moisture. Depends on the soil, like how well draining it is or whatever.
Yeah, when you're
This will be like on a scale of 0 to 100% saturation. And Dalton, were you saying something?
Oh yeah, that just answered my question. I was a little bit confused on what you meant by soil water content. I thought you meant like the amount of minerals or nutrients in the water in the soil, but you mean...
Oh, yeah. I'm talking about like how wet the soil is.
Okay.
Yeah. And you can either have 0, which is actually kind of hard in soils, or 100% saturated. You can't get more than completely saturated, right? No matter how much you add. Okay, so which of these are categorical, which are continuous?
So
Plants and treatment are categorical. Water is continuous.
All right, so continuous.
Sorry, Dalton. I interrupted you.
you're good, Trevor. You got the answers.
And then our dependent variable, we'll go with mass in grams, sure. Dry mass of plant. That is continuous, right?
Yeah.
Okay. You are very likely to be in a position where you're designing an experiment at some point in your undergrad or professional careers. You need to think about all these things ahead of time. Okay. Many is the experiment that has died after the experiment's been performed because it can't be analyzed because you set it up wrong. So let's think this through. We essentially have a pretty simple experiment here. We got 3 different plants, so 3 levels of plant, 2 levels of ag treatment, true or false, and then water. How are we going to do that?
You could vary soil types, like you said. So like maybe some like a peat moss base or something inorganic like sand versus like a larger granule diatomaceous earth kind of soil.
Now I got a problem with that. Can anyone anticipate it?
But introduces another variable of soil type.
That's another variable of soil type.
What about maybe a granule size of soil? So it's all the same kind of soil, just different granule sizes, so it adheres differently to water.
I've got the same problem with that. Granule size is a new variable.
Gotcha. Yeah.
So
Could
we're
you
just gonna
water one more than others and
we're gonna
measure
have to water one more than others. Here's the thing: ahead of time, we're basically gonna have to figure out how much water equals what percentage saturation in this soil. So that's a separate little experiment, but let's assume that we know that. And so let's assume that we can control just by the amount of water that we keep watering it at. So let's say we're not actually going to be able to do a complete continuous deal here, right? We're not going to be able to have 0.1%, 0.11%, 0.111%. We don't have infinite resources. So let's pick a few. So this will be continuous, but we're doing it at 10, 20, 30, 40, 50, 60, 70, 80, 90, and 100%. So how many levels of water do we have?
10.
Hmm?
10.
10. Yep. All right. With all that in mind, we open up Excel. Now, let's make this experiment. All right. Walk me through it. What's our data frame look like?
So you'd have a row for plants, right? And then, or not, sorry, a column for plants. And then you'd have a row. In the rows, you'd have plants A, B, C, similar to species, Ceratosa. And,
Like that?
yeah.
Okay.
Same thing for ag treatment would be a column, and then the rows would be true, false. And water, you'd have for a column, or water saturation, then you would put what, 10, 20, whatever, all the way down. And then mass as a column, and yeah, whatever the mass is in grams.
All right, and we won't know that until we actually do the experiment.
Yeah.
Okay. So we've got our column names. How are we actually going to set up this spreadsheet? Because it's wrong right now.
Well, you'd have to have an A true and an A false.
We need an A true and an A false.
And then inside of each A true, you have to have A true 10 through 100, A false 10 through 100.
Uh-huh. Okay, so let's think about this. We've got that. I'll just copy and paste that. Come on now. We want all those to be true, and then these to be false, and then water saturation 10, like so.
No.
All right, what's
So
wrong?
there has to be 10 rows that say A true. And the only difference between each one of those 10 is 10, 20, 30, the water saturation.
Right.
And then so forth. So you end up with a lot of like 600 or 60 lines. I don't know.
Like that. So we've got A true 10, A false 10, A true 20, A false 20.
That makes sense to me.
Okay. Now, currently the problem here is that we have replication of 1, which you can't do any statistics with. And plotting this is going to look really stupid also. So we have plant A with the ag treatment at water saturation 10. But that only occurs once in our entire data frame. So what's our replication going to be? How many replicates do you want of each plant?
3.
Okay. How are we going to show replication in this data frame?
We could do like A_1, well, I guess we would have to replicate that whole thing 3 times, but just add numbers onto each of the plant names that we have right now.
I see what you're doing, but I don't like it
Yeah, that's
because replicate is actually its own variable.
Okay.
So a trial column.
So insert column. Oh, it's getting pretty big. All right, so what we've got now, and keep in mind this is just for the first 2 water saturation values. We've got replicate 1. This is all replicate 1. That's replicate 2, and that's replicate 3. What are your thoughts about this data frame? Would you give this to a colleague, another peer working in your lab, and say, here's the data frame, I want you to collect the data and fill it out? Would you be comfortable?
you gotta do that thing that you showed us where you lock what they can put in.
You really do. You have to do that because they're going to say, oh, this one weighed 3.5 grams. This one weighed 3.4 grams. This one weighed 3.52. This one weighed 4.1. This one weighed 5. What's wrong?
They added commas.
Not everything's an integer.
Just ruin everything.
Typos. Yep. To err is human, right? So we want to prevent all that. So let's see. We're going to need all this, but again with 30, 40, 50, 60, 70, 80, 90, and 100. But essentially, we need to go up here, highlight this whole thing, and do data validity. All right, that brings up this. Allow. We'll allow decimals. And we'll allow empty cells. Okay. So if I do 3, 4, it says no. Okay. Another problem you might have, or I should probably say you will have, is somebody going down, entering stuff, invalid value, wait, decimals, data, validity. Did I do this wrong? Okay. Somebody's going through here. What happened?
They
Just
moved.
entered data in the wrong column and overwrit.
Yep, they moved.
Uh-huh. The finger slips, you hit the left button, and suddenly you're destroying everything. All right. So not only can you do data validation, or should you do data validation in a spreadsheet, but you should also highlight the rows that you don't want anybody to be able to change, and you can protect it. Where is that? Sheet. It's been a while since I did this. Does anyone remember?
Might have to ask good old Google.
I'm gonna have to ask good old Google. Tools. Protect document. Format cells, cells protection. Okay. Format cells. There we go. Cell protection. Protected. Okay. Format cells. Cell protection. Not protected. All right, so they can enter data here, but not over here. And then we go to Tools, Protect Sheet. You can give it a password or not, so you can just say, okay. So now, if I try to enter stuff in there, it says no. All right. If I try to enter stuff in here, it works. So when you're designing a data collection worksheet, which you will be doing at some point, keep this in mind. Okay. What if we over here had, oops, data, tools, unprotect sheet, date. All right, you might want to have date collected. If time is a variable you're interested in, you're definitely going to have dates. What's today's date? October
12th,
12th.
2020.
October 12th. Okay. 10, 12, 20. Does anyone know what's wrong with this?
It looks like math.
It does look like math.
Is it treated as a string though? In R?
No, it's treated as something really, really horrible by Excel. Let's see here. Format, cells. So if I turn it to text, which is what R is going to try to do. Oh, what's that? Four 4116.
Is there some
Is
sort
that the number of days from a certain date?
It is the number of days since January 1st, 1900. That's how, for some reason, Excel stores dates. It can't help itself. It must do it that way. So when you convert that into text, it just changes it into a number. So if you try to upload Excel dates into R, they get read in as this because that's actually how they're being saved by Excel. It's just pretending to show you 10, 12, 2020. In reality, it's storing it as 44116. This gets even worse. If you're on an older Mac computer, it's not stored as the number of days from January 1st, 1900. It's stored as the number of days from January 4th, 1904. Can anyone guess why? Anyone have any thoughts on this? I've got a hunch. I think that Microsoft was secretly working with the Russians to overthrow America, and that this was part of their plot, was by making us all learn to use Excel and have it destroy our data. In reality, there was a bug in the software for the Mac version, and to fix the bug, rather than actually fixing the problem, they just decided to change the way the date was stored, which screws everybody over for all time, okay? In this week's reading, we've actually got some resources on this. Where was it? Oh, it's in previous week's reading, I believe. I've got some resources for you I want you to look through about Excel, because as a data analyst, as a scientist, you need to know this stuff, okay? Excel is responsible for basically rendering about 1/3 of all molecular biology papers unusable. That's thousands. Countless hours of work and experimental money. And since they use Excel to store stuff, there's a problem. If I say gene oct11, which is the name of a gene, it may get stored as that, and it may not. If I then change this, oh, did I already set it to, oh, good for me. Format, cell, text, okay. Oh, here it was, oct11. It's gonna change things for me. Oct11. It always tries to change things into dates. What happened is that people were storing their data with the gene names. CEP22 is another gene name. It's getting converted into dates, which is then getting further converted into this numeric format. And if we don't have a record of exactly what version of Excel they were using and what type of computer, then we have no way of knowing how to turn that number back into the gene name. Okay. There's a news article. This is recent. Excel is indirectly responsible for the actual deaths of people in Great Britain because they lost 16,000 COVID tests. Any guesses as to why? No guesses?
Well, it says there's a 1,000,000 row limit, so they just ran out of. Excel can't take any more data after that, I guess.
This was
Probably
stored
deleted.
as CSV. When they opened it, that CSV file using Excel, it just silently cut off all the columns that it didn't want to handle. It didn't say I'm cutting these off or anything. It just deleted them. Okay. So row limits, the fact that it changes things into dates, you just need to be very aware of this. Excel is very good at what it is good for, and it's very bad for data analysis. So use it for its strengths, be aware of its weaknesses. Right? So some of the resources I have for you in here kind of walk you through how not to use Excel. All right. One of the things you should never do is have multiple sheets. And you'll see this a lot in people's data, and it's horrible, right? They'll have one variable in sheet 1, another variable in sheet 2. Okay. I encountered that kind of data in the wild constantly, right, from collaborators I work with, and it's really difficult to parse those. All right. Another thing you should never do is embed a figure in Excel. Nobody cares, right? It's going to look ugly anyway. We got ggplot. Don't put figures in Excel. Don't use formulas to calculate things in Excel. Don't worry about any of that. Treat it as, since you're data savvy now, essentially a data entry vehicle that does a really good job of that, unless you're talking about dates. So how do we get around this date thing? The safest way is to split it up into columns. Year, month, day. Okay. So we can just make sure that everything is treated as a year, a month, and a day. And that's going to be a number, and it's going to, R is going to be able to then make a date out of those really easily. All right, so I don't really want to go through and do all the copy pasting. So I'm going to go into R. And I'm basically going to use R to make my pattern data. So just a new window in Data Course on. I'm going to show you some tips for doing like repeated data, making Excel spreadsheet kind of stuff. Okay. So library, tidyverse, even if we're not going to use it, I just always load it out of habit. Okay. We've got replicate, plants, ag treatment, water saturation, and mass. Replicates. Okay. We know that that's going to be 1 through 3. And they're going to be repeated 1, 2, 3, 4, 5, 6, 7, 8. How many times? 12 times. So 1 is going to be repeated 12 times, then 2 and 3. Okay. Oh wait, sorry, 6 times, because we're doing it for plant A, ag treatment true, water saturation 10, B true 10, C true 10, and then the false version. So that's 6 times. So I'm going to repeat. Okay, so rep is a way to repeat things. And I'm going to repeat 1 through 3. I'm going to do each one 6 times. So that's 1, 2, 3. Okay. Is that, or is that not enough replication for our whole project?
That's not enough, right?
How
Because
many
you
do
have
we
to have
actually need?
10 times that, because you have 10 different water saturations.
10 times that, because we have 10 different water saturations. So 60. Okay. So I'm gonna cut that out and put it right here. Replicates. So we got 180 of those. It's 1 through 3, each repeated 60 times. Okay, my ag treatment. What's that going to look like?
Three trues, three falses, three trues, three falses. 180 times total.
Okay, 180 times total. So let's see. We could do. Is it going to be 3 true, 3 false? Actually, if we have it set up like this, we've got 60 ones in a row.
Oh, that's right. So it's 30,
30 of true, then 30 of
30
false.
false. Yeah.
So repeat true 30 times. And then we want to repeat false 30 times. And then we want to stick those together. And that didn't quite work. Didn't get us there. What's missing? That gave us 30 true and then 30 false.
You have to repeat that whole thing 3 times, right?
And we got to repeat that whole stupid thing 3 times. So I'm going to say repeat all of that 3 times. Now we've got 180. So we got 30 true, 30 false, 30 true, 30 false, 30 true, 30 false. Okay. And then how about our water? So we've got replicate 1. So that's basically the entire experiment right there, that first 60 rows. Okay. So we're going to be working in chunks of 60. Okay. Here we've worked in a chunk of 60. Here we've worked in a chunk of 60.
So these are 6 repeats of 10 numbers.
So we've got 10, 20, 30, all the way through 100. So that's 10 numbers. We want 6 repeats of each of those. In what order? Do we want 10, 10, 10, 10, 10? Or do we want 10, 20, 30, 40, 50, 60? 10, 20, 30, 40, 60. So we've got in replicates 1, our first 60, we've got ag treatment. All of those are true. So 30 trues in a row, and then falses. And that's all repeated.
So is it? Never mind. I'm just gonna be quiet.
Let's see here. So we're going to concatenate 10, 20, 30, 40, 50, 60, 70, 80, 90, and 100. Okay. Can we just repeat that 6 times, like so? Or should we repeat that each equals 6?
the second one each equals 6, right?
Let's do that. All right, and what's left? Plants. So we're going to stick together A, B, and C, and we're going to repeat that. How are we going to repeat A, B, C?
In that same order, ABC 60 times.
Okay. So plants, we've got ABC, ABC, ABC 60 times. So let's take a look at this. All of our columns have 180 rows. Things are looking pretty good so far. So we're going to make a data frame out of it. So data frame, replicate equals replicates. Agricultural treatment equals ag tert. Water saturation equals H2O sat. Plant species equals plants. Let's take a look at this. Okay. We've got true 10A, true 10B, true 10C, true 10A, true 10B, true 10C. That can't be right. What's wrong?
Don't some of those need to be false in order to be different?
They do need to be false. So we're going to have to do true, 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 false, false, false. True, 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 false, false, false. So the way we did our true, false isn't working. Okay, so let's think about it. We want to repeat true 3 times and repeat false 3 times. When you want to stick those together, like that, and then we want to repeat that how many times? 60.
Isn't it 30?
Yep, it's 30. That's better. Okay. So ag treatment, I'm gonna go in, replace that. Now let's look at it. True 10A, true 10B, true 10C. False 10A, false 10B, false 10C. All right. There we go. It is ready to rumble. I'm just going to add one more thing. Plant mass equals Na. So we're going to have a nice big Na column in there. That's the one we're going to fill out for our experiment. So now how do I get this out of R and back into Excel?
Right, CSV.
Just write a CSV.
Yeah, let's do that. All right, so I'm just going to send it into data, designed experiment.csv. All right, there's a few things I can say here. Row names equals false. I don't want it to add another column for me automatically. Quote equals false. I don't want to put quotes around anything like A, B, and C. And I'm going to have to tell it what to write. I forgot about that. DF. Okay. Did I spell experiment wrong? Yeah. That's better. Okay. So now here back in Excel, I can just open it up, go to desktop. Where did I find it? Data course is on. Data, designed experiment spelled correctly, .csv. Open it up. And there we go. Okay. So now I've used R, just a few little repeat tricks to make myself a nice experimental design. Okay. What do you think I'm going to do with this as your instructor? I'm going to generate a bunch of fake data and we're going to analyze it together. Okay. So this evening, I'm going to make a bunch of fake data for plant mass. And Wednesday, we're going to go through analyzing this experiment. Okay. The benefit of us designing it, being data savvy, is that it's already tidy. It's clean. It's ready to go. Okay. This is the rare, exciting time when you actually get to design the experiment as the person who's going to analyze it. You cherish this moment. We're just going to be able to read this in and immediately start making graphs from it the way we've written it up. Okay. I was kind of hoping you'd make some mistakes so that we'd have to clean it, but this looks nice. Okay. Another disappointing success. So Wednesday, we're going to analyze this together. Okay. But what I want you to think about right now, as a data analyst, what is it we're testing and what figures do you expect to see? What are they going to look like? Can you describe them to me? Anyone? Like, what's going to be on the
Let's see,
y-axis? Yeah.
maybe a dot plot with the y-axis being plant mass and the x-axis being water saturation, maybe colored by species.
Great. All right, that should be pretty easy to do, right? That'd be a ggplot where we do df, our aesthetic, x equals plant mass, our y equals water saturation, color equals plant species.
As a general rule, you'd put the plant mass as y, right?
Yep,
Versus
Sure
what
would.
is saturation. All right.
I messed that up. I'll just change those. They can be out of order, that's fine. And then geom point and geom smooth. All right, we can already plot that. That's great, isn't it? Well, they all are NA right now, so that's fun, but okay. What, yeah, replicate will be fine because we got 3 values for each one of those. That's totally cool. Bingo. So that'll be a nice plot right there. What about a statistical test? Has anyone had stats or is currently taking stats?
You do like an F and a T test.
Checking it right now.
Sorry, both of you spoke at the exact same time. I couldn't hear either one.
I just said you could do an F and a T test
Okay.
based on trying to find differences between species or between saturation levels.
Right. Sounds good. All right. Well, I'm not going to get too into it right now. But fall break is until Thursday, right? We do have class Wednesday, right?
Yeah.
Okay, good. Wednesday, we're going to be starting statistical modeling, all right? You already know how to import data. You can do some tidying on it, make it ready to plot. You can make pretty plots and really hideous plots. I've seen your work. And next on our data analysis lifecycle is modeling. So be ready for that. If you would like to prepare, which I would recommend, I would go here to week 9 and take a look at what is the stat model, modeling intro, basics, and model fitting. Okay, look through those. It's okay if you don't understand everything that's happening in those, but I want you to be familiar, especially these 3. These are chapters in the R for Data Science book. They have really nice walkthroughs with pictures that kind of show you what we're going to be covering. Okay. I am essentially putting off the week 8 stuff, writing functions, conditional execution, sourcing things for later, okay? Because this is already midterms, you've got lots of exams coming up, including one in this class, which is open now, by the way, and which is due before next Monday. So we're going to kind of save that and move straight into the modeling stuff. Sound okay with everybody?
Yeah.
Okay. In light of today's kind of big picture thing, we also, if you remember from the syllabus, have a final project in this course. Anyone remember that?
Yep.
Well, you do, yeah. Your final project is going to be an entire data analysis from start to finish. So finding the data set, importing it, cleaning it, visualizing it, modeling it, and then presenting it. Okay. It can be anything you want. It can be data for another class. It can be data that you're doing as a research project. All of that's good, but I need you to identify by the end of this week the topic that you're going to do your analysis on. Does anyone already have stuff in mind? I believe Lacey and Trevor, are you going to take me up on that?
I was just gonna ask, is that does that count for the project as well?
It's a data analysis task. Yep,
All right.
counts for the project.
Perfect. That's what I was
Nice to have
planning
one
on.
then.
Okay.
Wait.
So as my research students currently, those 2 are going to be working on some metagenomic data from mangrove forests, mangrove swamps in Indonesia. All right. So that's a research project. Anyone else have stuff they're interested in? It does not have to be biology. It's fine. It can be for another class. You can. I've had students try to predict baseball games based on batting averages of players. I've had students look at police stop rates for different genders and ethnic groups across the country. Students doing GIS mapping through R, looking at like salmon reproductive potential in different streams. The sky's the limit here. I can help you find data, but only if you get me your topic real soon. So now's the time for wild ideas. So bring them on. What are you interested in? I'll tell you if it's feasible, if we can find data sources for it.
Quick question.
Yeah.
By next Monday, do you mean the 19th or the 26th? For
A week for the exam?
Yeah.
A week from today. nice.
Also with the exam, I just did a poll on the data course repository, and I don't see it in there. Where would it be?
It's a separate repository. It's the exams.
Oh, okay. Sounds
So
good. Thank
if you
you.
go all the way up here, right here, the exams GitHub repository.
Do we
So
also have an?
make sure you clone that and get it on your computer. It's pretty straightforward, this
All right,
one.
sounds good.
It's a little cleaning and tidying, mostly ggplot. Okay. No functions are actually on it since we haven't covered that.
Do we also have an assignment, or is it just the exam
Nope.
that you're pulling Monday?
Just the exam.
Nice.
All right, so who's got a crazy idea? What do you want to research?
Could you give me some sort of bioinformatics, like genetics data?
Like what kind?
Let's see here. Could we do?
Preferably it's something you're actually interested in because you're going to be spending a lot of time with this data.
Yeah.
Dr. Zan,
Yeah.
what is the end goal for that project? Like just produce a graph or?
No, an entire analysis. Let me show you. Oh crap. Not on this computer. Some previous students' work. Essentially, you're going to be posting it online like this, and I can help walk you through that. Let me show you Markdown. So you're going to be describing your project and incorporating your code into basically a reproducible report. Okay. Okay. So something like this.
Whoa, let's see.
All right. So this person has incorporated all their code into a report, so you can generate a website or HTML document, a PDF, or a Word document just by running some R code. All right. The idea here, HTML widget, Shiny, dashboard, presentation, books, websites. GitHub, CB, all. HTML preview.
GitHub.
All right, here we go. So this is an example report. yours probably won't be this extensive, right? This is for an actual published paper. But you can see how he's got an explanation of the background of this project. Okay, so like why you're interested in this data set, essentially, what we know about it. And then here he's describing it. He's included some photos, a nice little image here of his site. Okay. He's talking about the methods that were used, describing the data that he found, showing the parameters, dissolved nutrients. Here's the ANOVA tables for his statistical tests. So that's displaying right in line here. And then the output. Okay. So it's basically a scientific paper, but just the analysis part. Okay. Yours won't be this long and extended because you don't already have a PhD, and I'm not expecting you to make a publication quality thing, but just to give you an example of what's going on. This is R Markdown. It allows you to incorporate text and your analysis. So here he's got a little button code. So if you wanted to see the actual code that he ran to get this result, it's built in. It's just kind of running behind the scenes. So the end goal for your final project is to have a report in HTML format that we can throw up on the website that describes your data set, why you're interested in it. It does your cleaning, and it shows off some of your nice plotting skills and comes to some conclusion about your data. So in essence, it's like a short scientific paper that you're writing.
Okay.
Definitely with me grading more heavily on the cleaning, data presentation, and stats tests than on like whether it's going to be publishable or if you included correct citations or anything like that. I just want to see your analysis of the data set.
Okay, sounds good. Thanks.
So who's got ideas? Dalton, you were saying genomics of some sort?
Yeah.
Kind of a big,
Broad.
yeah, it's a bit broad.
Can we do something on inducing genes into bacteria? Is that specific enough?
That may still not be specific enough. What kind of data would that be? Like whether it worked or not? Like what conditions?
Yeah, that's the problem is it wouldn't be very good data. It'd be pretty much what happened.
Yeah.
I got to keep thinking.
Okay. Who else?
Quick question. How are we going to get our data in the
We're
first
gonna
place?
Find it online. I can help you with that.
Okay, I kind of have a funny idea
Or
from something
if you've got your own data.
from
but,
something
yeah.
I heard. What about rodent populations and house fires? Those two might go together. Well, because I once heard that they did, but I kind of want to look at that one.
Oh, well,
Or like mice and rats in particular.
yeah, that might be hard. That data set doesn't exist as it is, I'm sure. So you would have to find data sources for like house fire incident reports online, and in the same locations, like population models for rat and mice, and combine them, right?
Or it might be easier to find something like reported infestations or something.
Yeah. The problem is going to be finding that data.
Maybe I should do something easier.
It's possible. It's possible. Maybe. A couple semesters ago, I had a student who was really into like predatory birds, raptors, and so there's an organization called Hawkwatch, and she wanted to look at like predatory bird survival across the world and basically make a map correlated with climate variables, which is a cool project. Hawkwatch agreed to send us some data after much back and forth, and they never did. I mean, we're talking like 2 months. They never sent us this data, and then a year and a half later, they still haven't sent it. So finding data can be an issue. A lot of times we run experiments just because we can't find that data, right? Like, oh, we're going to have to do that experiment. Let's go out and torch a bunch of people's houses and then count rodents afterwards. I'm kidding.
Brilliant.
So for like genomic type stuff, metagenomic stuff, a lot of that's going to be widely available. We can go to SRA, Sequence Read Archive, like NCBI, and get genomic data sets. Okay, so we can search for those like by species or whatever. Organism. Yeah, I spelled that wrong. Okay, so we can get RNA-seq data from mouse studies, right? Those are widely available. We can get human microbiome data. We can get gene expression data. All that kind of stuff is going to be available on here. Genomic stuff does take extra steps, right? It doesn't come in a two-dimensional data set. This is raw DNA sequencing. Okay, so if you go this route, you're going to have some extra processing steps that aren't necessarily going to be in R. You might have to break out some command line tools to kind of get the data whipped up. Other times we may be able to find other stuff for you. At any rate, it doesn't sound like a lot of you have ideas for what you'd like to learn how to analyze, but I want you to by the end of the week. Is that all right?
Do you have office hours that I can maybe I can talk to you about?
They're not set office hours, but I do have availability pretty much every day. So if you want to send me a note with like propose a couple times, that'll usually work.
Is morning better?
Morning is better
Okay.
than afternoon. Yep, I have more freedom.
Yeah, I'll send you a message probably tonight, and then maybe we can meet tomorrow.
Yeah,
Okay.
that'll work. Yeah, some of these conversations may have to be one-on-one because all of you are going to pick pretty wildly different data sets. I'm aware of that. The stats student I had a couple years ago who did baseball stuff, I had no idea how to get baseball data, so he had to go find it on his own. But that was very different from the genomics data that other people were doing. The common theme here, though, is that everybody's importing, tidying, visualizing, and testing data, and all the modeling essentially looks the same at the end. It's just a matter of the cleaning steps. Baseball data comes in a very different format than RNA-seq data from a mouse liver. So be thinking about that. Think real hard. I really would like, by the end of this week, at least your topic, maybe not your perfect like designed experiment that you found and you've got data for it, but at least a topic, okay? Try to keep it something that's doable, and keep in mind, if you do pick genomic stuff, you're gonna have to spend more time on it than somebody who's looking at like police reports. Okay, so some of that may influence what you decide to do. I just want you to kind of be aware of that. All right. Any questions? Today was a bit different from class as normal.
No, thank you.
All right, you're free to go. Definitely go through the assignments, keep practicing. Look at the modeling stuff before class on Wednesday so that you're not super lost when we're going through it. Because Wednesday I'm going to be not moving light speed or anything, but not stopping to explain the big picture about everything as we do the steps, so that you just get kind of this experience of, we designed a data sheet, now we're going to analyze it and see what that looks like. Okay. Then next week, after fall break, we'll really get into the nuts and bolts of what modeling is. Right? I just want you to see it first. All right. Well, I'll stay for a few minutes if people have questions, but you're free to go.
Thanks, Dr. Sun.
See ya.
I have a question
Sure.
about a potential project. So I'm working on my senior thesis right now about possible connections between autoimmune diseases. So I was wondering if I could potentially look at genes that, so do my data analysis on genes that are related to multiple autoimmune diseases. Would that be a possibility?
Yeah. Do you have a data set in mind already, or?
I have one person who already did. I have one article that already did a meta-analysis.
Okay.
Of it. Should it be okay to just use the one that they did, or would it be better?
Yeah, absolutely. No, I think you can. Redoing somebody's analysis is a very good learning exercise.
Okay.
So I fully support that. Is their data set available?
That is what I'm looking at right now. I just need to find where that article is.
If not, you can always recreate it, because a meta-analysis obviously comes from a bunch of other studies. So you could find the data in those studies, recreate the data set that they use for their meta-analysis that way.
Okay.
But ideally, they made it available somewhere.
Okay, so I found it. How would you find? Would it just be at the end of their paper, probably, where they have their
What's
data
the name
set?
of the paper?
The name of the paper is. Hang on, I lost it again. It is. Okay, let's see if I can read this. Celiac disease immunochip consortium, rheumatoid arthritis. Or is this the company? Oh no, it's meta-analysis of immunochip data of 4 autoimmune diseases, reveals novel single disease and cross-phenotype association.
All right, say it again slowly.
Okay, yeah, sorry. So it's meta-analysis of immunochip data
of immunochip?
Immunochip, yeah.
Okay.
Data of 4. I can actually see what you're. 4 autoimmune diseases.
Got it. Reveals novel single disease and cross-phenotype associations?
Yep, that's it.
Okay.
Yeah, that's it.
Okay. So they've got some tables here in really horrible format. Yeah, all right, so their tables are available. It's just going to take some cleaning.
Okay.
It's awful. Sometimes there'll be a data availability statement in a paper.
Mm-hmm.
Availability of data materials. Okay, all data generated are included in the article and its additional files. So that's annoying. It'd be really great if they put this in CSV format for people to actually redo, but they didn't. However, that's okay. It's all to be found here. So we can download these files here. Summary stats, we don't want those. Results of the meta-analysis, no. Okay. Case control data sets used in the study. All right. So that's convenient.
Okay.
Here it is. I mean, you can see how terrible this is.
Yeah.
You would never be able to ggplot this, and like the numbers themselves have a and asterisk after them. So that's stupid. But here we go. This is a bit closer. They've included a nice merged cell up here with the name of it. We've got 2 header rows. All right, so R can't even properly import this. But there is the data.
Okay. So I just have to fix it all.
You would have to find a way to import all of this stuff into R. Maybe not even all of this. Potential role of... See, that's dumb. Region, chromosome position, SNP, 1. You may be able to find it in here. I mean, I don't actually know what this paper is really about, but yeah.
Right. Okay.
So it's possible.
I'll take a look. And then if
Yeah.
I'll probably have to set up a meeting with you a different time to actually try to import it.
Yeah, the importing is going to be hard, but so is understanding what all this is. This is obviously not a tidy data set, right? Each variable is not its own column in this. So I'm trying to figure out what it is, what A1 actually means. I don't know. Oh, you'd have to look down here, I guess.
Alternative allele.
Okay. Used in the logistic regression. Part of me thinks and fears that this is actually already summarized data. It's not the raw.
Oh. How would you determine whether it's the raw or the just... Do you think they'll mention it in the article?
Yeah, you're going to have to read this paper really closely.
Okay.
But if this doesn't work, I think that's okay because they got their data from these papers. So you could go to these papers and download the raw data itself.
Okay.
Whatever immunochip data is, or whatever it looks like, I'm guessing that's just some technology. They use the R package asset. So there's a lead for us. Subset-based association analysis of heterogeneous traits and subtypes. Okay, so we've got a package. You'd have to learn how to use this package.
Okay.
What I don't see in their thing so far is their code. So they've done a lot of work in here that's not necessarily represented in that data set that they gave us. I think it is summary. We performed a subset-based meta-analysis applying the H-traits function implemented an asset. Okay, where's their code? Right.
Right.
I have no idea. This is not 1935. We should be all providing code with our data set or with our papers now. It pisses me off to no end when papers do this, especially a meta-analysis. Like, we've got 400 pages of code that we went in for collecting all this stuff, and you can't see any of it. So I don't know if they did it right or anything. And I also can't learn from it. So I hate when I find this, but it is more common than I'd like. I don't think they're going to be sharing their code. One thing you could try is this person here is the corresponding author. So is that one. I would maybe try emailing this person in Spain and asking if they have a code repository for this paper.
Okay.
It's worth a shot. Yeah. Sorry, I can't be more helpful.
No
Uh,
worries. I
Yeah.
didn't know I found such a difficult paper.
Well, you've got, it's not a difficult paper, it's an opaque paper. They're not including their actual raw data. They've got what they're claiming is raw data, but it's actually summarized stuff. So who knows? Maybe you can find stuff in there. I download all of these files and take a look through them, see what is available.
Right.
Yeah. Yeah. So look through it, get a feel for it, and set up a meeting, and we can chat some more.
Okay. Thank you.
All right. See ya.
See ya.